In 2016, the J. Craig Venter Institute announced that they had generated the world's simplest independently living organism, the JCVI SYN 3.0. This is approximately what they did. They started by reading the Microplasma genitalium, or M genitalium, DNA into a computer. Then they extracted the DNA from an M capricolum and replaced it with the computer's DNA. They placed the new organism in a pristine environment. It lived. It reproduced. It had the body of one organism and the DNA of another. They accomplished this in 2010 and called the organism the JCVI SYN 1.0. This was a major breakthrough, as now they were able to make any change they wanted to the computer's copy of the DNA and see what happened. Once they had this technology, they embarked on a venture to make the simplest independently living organism. They deleted one gene from the computer's DNA image and put the result into the DNA-less M capricolum. If the new version lived, they concluded that the gene was unnecessary, at least for basic life. Then they tried the next gene. If the resultant organism did not live, they decided that the gene was necessary, so they undeleted the gene. They kept doing this, effectively playing whack-a-mole with the genes in the computer's DNA image. In 2016, they were done. They called their organism the JCVI SYN 3.0. It had 531,560 base pairs, 473 genes. 149 of those genes had unknown function. I decided to visualize this a little bit. I downloaded the computer version of the DNA. I then printed it as colorful bars, red, green for AT, blue, yellow for GC. By flipping the bars upside down, I was able to render a nice approximation of the DNA. I initially envisioned 3D printed Lego-like blocks, but the task proved ominous. I tried printing where each bar was 1/8 inch wide. Still, the results were enormous. It would print out at over 2,000 sheets. I settled on 1/20th of an inch wide bars. That was the smallest I could make and show the letters readably. I also created a 3-barred code to represent alphanumerics. This is similar to the 3 base pair amino acid system that DNA uses. I paralleled the DNA with the novel The Call of the Wild by Jack London. The results are printing along the bottom of the screen. If you pause the video at any point, you can see 1 page worth of JCVI SYN 3.0 and 1 page worth of the novel. So let's see how dense this code is. An American quarter covers 25 base pairs. A quarter is a bit thicker than a printed bar, so a stack of quarters representing this DNA would cost over $130,000 and be over 1,100 feet long. Let me address a major misconception that I used to have. I used to think that genes were tiny things. If we take 531,000 base pairs and divide by 473 genes, we get well over 1,000 base pairs per gene, or over 333 amino acids per protein. There is some required epigenetics in the organism. Typically, a gene is 150 aminos long, with 450 base pairs. So it appears that the genes in the JCVI SYN 3.0 are particularly large. So let's look at the volume of information in one gene and parallel it to the text. You can see that it parallels to about a paragraph of text. It contains the careful instructions to make one 3D part. Each 3D part is radically different from other 3D parts. Very often these parts work in conjunction with other parts to do a particular task. Now notice also that Venter says that about 1/3 of the genes have unknown function. However, without these genes, the cell dies. There is a lot more research to do just to understand this simplest organism. The DNA printed out on 1,068 landscape pages. Each page renders 10.5 inches of DNA. I got friends and family to help me tape these sheets together into a mighty bifold. We then found a quiet street and spread out the printout of the DNA. We placed rocks on every second or third page so that they wouldn't blow away. You will notice a fast motion video of the rollout in the upper left corner of the screen. By the time we got this monstrosity rolled out, light was seriously waning. In any case, you can see that the printout goes on and on into the black. Yes, the DNA required to make the simplest life form on Earth is, well, enormous. Some conclusions. This organism is the closest we have come to the legendary LUKA, the Last Universal Common Ancestor. There's something we need to understand about LUKA. Bacteria and archaea separated about 3.5 billion years ago. They share most of the same genes. Therefore, this technology was fully developed 3.5 billion years ago. So this is not the product of billions of years of evolution, maybe 100 or 200 million, but not billions. So all life that we know, whether in nature or generated by man, is just amazingly complex. This has been scientifically tested to be irreducibly complex at the gene level. Therefore, most, if not all, of the multi-gene subsystems in this organism are also irreducibly complex at the gene level. In each of our living cells, we have 3 billion base pairs from each parent. This encoding scheme renders 1,267,200 base pairs per mile. An entire listing of one parent's contribution to our DNA would print out 2,367 miles long. So if we printed it out, it would stretch from Anchorage, Alaska, all the way to Disneyland. Whoa. As I look at my next video, I want to address where science is really at regarding origin of life. Can scientists generate life in the lab? Dr. James Tour claims that origin of life scientists are clueless about how life originated. Is he right? With all of the headlines claiming again and again that finally life's origin has been solved, how can we really get a true answer to this question? I promise you, we will. Even if you are a non-scientist, you will be able to be confident of where origin of life research is at now and into the future. Please like, subscribe, comment, and share this video.